right, thanks Ian, and welcome everybody to the session. So, so for starters, I realize that probably not a lot of people have worked on diseases before, but I thought of giving this title for today's talk, because no matter which species you work on and which taxon, chances are that at some point in time you might encounter diseases or pathogens, and even if it's not a problem at the moment, it might be in the near future. And that's just the thing about diseases. If we're not prepared, or let's say we have to be prepared for the unexpected. So it is true, and if you look at literature and about conservation, that emerging infectious diseases is becoming a hot topic. It's being published more and more on, and especially for the last 3 decades, there's absolutely been, literally been a bloom on papers concerning infectious diseases with various taxons. So it. I've done it again. Right, so perhaps just a definition to start with. So when we talk about an emerging infectious disease, those are diseases that have recently expanded in their geographical range, or that have moved from one host species to another. So they've undergone, or they could simply have just undergone some kind of change in pathogenesis that also qualifies the pathogen as an emerging infectious pathogen. Right, and from a human health perspective, even though we're talking about wildlife diseases, it's also relevant, since health diseases, human health diseases, almost 60% of those involve some sort of zoonosis, or some sort of transmission from wildlife to humans, and therefore really relevant to human health. And the majority of those originate from wildlife diseases. And Ebola virus, of course, is one of those examples. Right, so emerging infectious diseases, whether it's of humans, from wildlife, or from plants, can have got a few things in common, and they can be characterized by the process of recent expansion. So there's some sort of a change that's happening within the life cycle or within the epidemiology of the particular pathogen. And secondly, these are often linked to anthropogenic change or environmental change, whether that be habitat destruction, whether that be increase in agriculture. It could be a change in human behavior, as with the wildlife trade, for instance. Right, and just to refresh our memories, a few examples of recently emerging infectious diseases. We all know West Nile virus that had an epidemic in 1999 in North America. And that threatened both human and wildlife at the same time, simultaneously, with over 15,000 people infected and more than 200 species of birds infected. So, and that's the thing with these emerging infectious diseases. They're often not very host specific, and therefore relevant to a broad range of taxonomic criteria. So Hendra is another example. An outbreak in '94 in Australia, for instance, when carried by flying foxes, not pathogenic. The moment it's transmitted to either humans or to horses, it does become pathogenic, and deaths do occur. And it actually became an economic problem in Australia, because it especially infected racehorses. And then more recently was white nose syndrome. That was, it's a fungal pathogen of bats. It's been around in Europe for a very long time, but recently emerged in the United States. And since then it's spread across the entire United States, just in a few short years. It's caused more than 7 million bats to die, and more than 90% declines in some species has already occurred because of this one single pathogen. And then these are the group of organisms that I work with, that my lab focuses on. Also a fungus, Chytridiomycete fungus. The disease is called chytridiomycosis. First emerged in the late 90s, simultaneously in the Australian tropics and the Central American tropics. And it's absolutely devastated amphibian populations around the world, and some species have even gone extinct in association with this pathogen. Right, and those are really just a few examples. But right, let's get to the meat of the talk, and that's about disease surveillance. So it is true that surveillance of disease outbreaks in wildlife are particularly relevant today, given this context of newly emerging pathogens in a range of different taxonomic groups, and also because of the increase in animal trade and translocations that have been occurring over the past few decades. Right, so it involves, per definition, thus, detecting, determining presence, and characterizing these different pathogens, followed up by subsequent monitoring. But that's basically what we're talking about. And it is true that countries that have been vigilant, countries that have conducted disease surveillance of wild animals, are simply just more likely to detect and promptly respond to the threat posed by these diseases than countries that are not. And secondly, delays in the detection of infectious diseases have also contributed to failures often on a regional, national, international level of controlling epidemics and epidemic outbreaks. Right, so pathogen surveillance, therefore, is normally the responsibility of government departments, usually departments of agriculture, and this is an international phenomenon. But unfortunately, it does not often include regular monitoring of wildlife, and that presents a problem for all of us in this room interested in wildlife and nature conservation. So internationally, there's a list of pathogens that are affected by international trade, and these are listed by the World Health Organization, Animal Health Organization. But it's only countries that are member states of the OIE that have to adhere to these guidelines. But recently there's been a bit of an improvement, and the early information system of the OIE has started. It's an online electronic surveillance system, and member countries have to report, firstly, outbreaks as soon as they appear and happen in real time, but also submit 6 monthly reports to the OIE. So that's progress in the right direction. And OIE listed chytridiomycosis together with a virus. So those are the only 2 amphibian pathogens that are currently listed by OIE as notifiable. And they also provide a set of guidelines for containing and preventing spread of these pathogens. So right, as far as chytridiomycosis goes then, this map shows the sort of international collaboration and international surveillance that's happening with this pathogen. So it's literally an international effort at understanding, studying, and discovering, and also notifying this pathogen. So there's a specific website where information can be posted by individuals, by organizations, and this is kept updated at a regular basis. And it really does provide the first baseline for conducting surveys, seeing what the context of a specific country is with the rest of the world or adjacent regions. Right, and that's what the fungus looks like. So these are laser microscopy images. So spherical fungi with lots of little zoospores, swimming zoospores within them. That's a little bit of what the pathology looks like. So it infects the epidermal skin, and it causes these specific lesions associated with the sporangia. And there you can see again, those are epidermal cells that are infected, and with little discharge papules through which the zoospores are then leaked outside and then transmit. Right, so in terms of survey protocols thus for diseases, and I cannot stress this enough, that it involves an absolute knowledge of the host organism, and that's part of the message today. If we work in teams and in groups with different expertise and combine different expertise, is that if you know the host ecology and the host biology, it's so much easier to start thinking about how, when, and where do we want to start surveying for pathogens. And we have to lead ourselves by the different geographical regions, by the different taxa that we work on. We have to consider life cycle. Do you want to focus on specific taxons? Do you want to focus on specific conservation criteria or conservation status? Or do you want to focus on a specific life stage or reproductive mode, at least, in the host organisms? These are all things that will be that can be advised by the different taxon specialists working in these teams. Right, and so once you've decided which species and which regions to work in, you have to narrow down those specific searches. So what you do next is to conduct risk assessments, and this is what we did for Madagascar with Stefan Lotter, and doing a climate niche. So it's GIS layers with environmental parameters as well as distributions of the different species. And once you produce these risk zones with high and low risk areas, you have to compile a list of possible monitoring sites, and there it really, again, it depends on the geographical region, it depends on the group of animals or plants that you work with. But for instance, for Madagascar, we considered where major import ports are, airports as well as harbors. We looked at where the specific manpower and expertise are distributed on the island. We looked obviously at this map, distribution map, high risk, and also included a few low risk regions. But that's all part of the planning phase of where you want to go and how you want to get at those sites. And then there's something really important to consider, and that is detection probability. And that's the difference between pathogens working with diseases and working with host organisms or primary organisms, is that they're often invisible, and we don't know where to, and we don't know whether they're going to be present or not. So you have to work with some sort of a plan, some sort of an estimate of where and how to find these pathogens. So when we started with amphibian disease research, we didn't really know how to sample in Africa and adjacent islands, and so we did what the fish guys did, because they've been dealing with stuff like trout whirling disease for many years. But they work on a system called the assumed pathogen prevalence level. And they assume for fish diseases a 2, 5, or a 10% prevalence within populations. So that was really our initial baseline that we worked with, because proper chytrid or chytridiomycosis surveys obviously relies on an underlying knowledge of prevalence within those populations that will be sampled. And so US Geological Surveys first then suggested that APPL for chytridiomycosis should be about 10%. And that's where we started. And where this 10% comes into play, or prevalence comes into play, is that determines the number of animals that need to be sampled in order to yield negative results and be 95% confident that those negative results are reliable, therefore that the pathogen is not present within that specific geographical region or population. I'm not going to go into any specific details here, but there are various models, there are various equations available for working with that presumed prevalence and thus determining sample size. And you can clearly see as predicted prevalence becomes lower, there's an almost exponential growth in the number of animals that need to be sampled. So these are things that you're going to need to consider no matter which taxon and group you're working with. And then another aspect is thinking about the biology of the species that you're working with, because often pathogens present themselves differently in different life stages of host organisms. With amphibians we found, and this is a study that we did in Montes Sources and down in Royal Natal. So we've got river frogs up in the berg, and we've got ghost frogs down in the valley, and the exact same pattern emerged. So we've got low infection in animals that are in newly developed or in young individuals, and then we've got much higher prevalence in more developed and larger animals. So even within a single life stage, if there's different developmental stages present, you can get differences, and you need to know these. This needs to be considered, because remember, you want to maximize the chances of finding the pathogen during these surveys. Right, and the next important thing, of course, is which diagnostic tool to use, and of which there's often lists of different tools that you can use. So you're going to have to decide whether you're going to use a tool with high specificity but lower sensitivity, like for instance histopathology, or whether you want to follow the course of high sensitivity and use molecular techniques and molecular tools. And it really helps if research has been done on those particular tools and there's scientific backing as far as comparisons go between the different tools. And again, I'm going to go really quickly here. Of of course, once it comes to diagnosis, the very first thing that you need to know is know the clinical symptoms. Again, sometimes it's easy, sometimes it's a specific disease-specific symptom, but often, as is the case with amphibians, there are generic clinical symptoms, and it's often hard to just go by this. So you really need to go further. This is just a lesion. I'm just going to go through this quickly, going deeper into the skin. And this is just a wet mount of amphibian skin. Start seeing a lesion opening up, and as you focus deeper to the lower parts of the epidermis, you start seeing the actual sporangia and the pathogen appearing. So you need to know where to look and how to find the organisms. Histopath is really handy. It really tells us what's happening. This is the interface between the pathogen and the host. And once you start understanding how it infects, what it does, what the host response is, you can learn a hang of a lot. You can also do retrospective surveys. And by doing that, find out when the pathogen first appeared within a region and how it's spreading from those loci. All right, I say I'm running out of time quickly. This is obviously just more advanced diagnostic techniques, including sequencing, learning a lot about different strains, and that's something that will have to be considered as well. Is the pathogen you're working with really just one species, or are there differences in different strains and lineages? So has there divergence taken place or not? Because your tools might not pick up all of those. So once you've done your predictive modeling, based on the true data, you have these different risk models. Combine that with the international networking groups, and then we use all of these information to start mitigating. And this talk is not on mitigation, but basically there's 3 different international projects here, and they were all based, completely different methodologies for mitigation, but they're all based, and that's the commonality here, on initial surveys that were done thoroughly, that rendered information on which to mitigate. Right, and this is the take-home message in the final slide then. And it really talks about conservation medicine, and this is a concept that was proposed a few years ago by Peter Daszak and colleagues, but it really talks about taking a novel approach of having interdisciplinary teams working together. It involves conservation biologists such as ourselves, ecologists, people from the medical sector, veterinary as well as human health, working together, looking at it from different angles, environmental, animal and human health perspectives, and only by combining these teams can we have the necessary abilities and resources often to tackle these issues that are on the rise. Right, and that's my lab. I'd just like to thank all of them because this research is really a team effort from the guys in Potch. Thank you very much.